This is Katie.
And I'm Vinny, and you're listening to Learn Real Good.
Wow, I'm jazzed.
Why are you so jazzed?
It's been a while, actually. There was a rush when we were recording like a bunch of episodes in a day, and like it's been a while.
Yeah.
It's good to be back.
It's great. Our audience won't notice because these come out in theory weekly.
I know you don't like me alluding to any current times. I'm always careful not to mention, summer weather or the Olympics like I just did, because we don't want people to know.
No, this could be
this
recorded.
Could be Christmas.
Yeah,
in fact,
maybe
maybe
it is Christmas Eve.
Yeah. But the Olympics happened this year. That's true.
True statement. The 2020 Olympics happened this year.
That's already confusing.
Yeah.
Right? Because they weren't able to occur because of the obvious pandemic last year.
Well, they're not going to redesign all the merch.
They have all those t-shirts.
Yeah, they printed up a lot of logos.
I mean, going from a 0 to a 1 is a hard one too. Like if this was going from
1 to 4, that's an easy fix.
1 to 4, yeah.
1 is an easy. 1 to 4 is an easy.
2 years.
Yeah.
3 years.
Yeah, 2 years then another year.
Right. And thank you. That's what I was going to say.
Yeah.
That's how I measure time.
Yeah.
That would have been easy.
Easy fix.
They were unlucky there.
Very unlucky.
But I've been enjoying them. I always love Olympics. It's so exciting.
It is exciting.
And it makes me think to tie this into science, because this is a science podcast.
Sure.
Just the physiological feasibility of some of these feats.
Well, I mean, you were telling me how the 100-meter dash, they only run for 10 seconds, so the cells draw on a different source than an endurance runner would. So those first 10 seconds of physical exercise is consuming a different source of energy in the body.
Yeah, and what's interesting with that is that I only recently sort of made that connection, because there is this process where your body gets quick energy in the first 9 seconds is how long it typically lasts in the average person. I don't know if it's different if you're
maybe if you're world-class athlete,
but I imagine it's a bit of a physiological limit. And so it's cool that lines up perfectly. I imagine that's not just coincidence, that they really have that length of race to sort of exploit that one source of energy.
Mm-hmm. That's how they should measure the 100 meters. So how long before you switch your cells to another source of energy?
Yeah, I think that's hard to measure. It's not something you notice. You do notice your aerobic system kicking in. That's sort of when you hit the wall a few minutes
9 seconds in is when I hit the wall.
Right. Well, we are not Olympic athletes. We are scientists and comedians.
That's it.
And that's what makes us well suited for this podcast and not the Olympics.
No, not
just
the Olympics.
Fans.
Yeah.
They need fans too.
Yeah.
Oh, gosh. Are you ready for our facts? So for new learners out there, we do have a guest and we're excited to get to them. But before that, me and Vinny share facts.
I love facts.
That we recently learned. Are you ready?
I'm going to go first.
Oh.
Because I enjoy my fact. This is a good fact.
I mean, I don't want this to be a fact off again. I also
we get pretty competitive with our facts because of the Olympics.
Well, yes. We'll make the guest give us gold and silver in best fact. And part of it is delivery. Part of it is the quality of fact.
You have a whole criteria system set up.
Yes. I'm jazzed. Okay, let's compete.
You do love a good competition.
Representing Canada.
Both of us.
I know.
So already the country's going to get gold. That's
yeah. Win-win for
Canada.
Win-win
for everybody.
We love that. Go for it.
All right. So my science fact is purely a measurement achievement. It's kind of cool. I love measuring things is very complicated. And you're laughing at me already. But measuring things is a whole field of physics of how heavy is a kilogram? How do you know how fast the speed of light goes? These are questions that are kind of fundamental to the universe. But this is an engineering measurement question. So NASA, a couple years ago, sent a box into space. It's just a small little box, relatively speaking. It's the size of a microwave, I'd say.
Okay.
And it's a clock. That's the whole thing. The whole thing is just a clock.
Wow.
It's an atomic clock, and they sent it into space, and it's just sitting up there keeping time and beaming the results back down to Earth. And on Earth, we have very precise clocks. They're super, super accurate. Each country has its own national clock. They're not synchronized necessarily.
Wow.
And so they generally try to keep everything in universal coordination, but each country is responsible for its own timekeeping units. Just like the GPS is an American run system, and they used to have it, only the U.S. military would get the full precision of it. So the U.S. military would be able to detect themselves with 1 meter accuracy, but they would kind of skew, fuzz it up for 10 meter accuracy for for civilian use or other countries. I think they've since dropped it because you can just use software to kind of, if you can get the signal, that 10-meter fuzzing is negated by more measurements. In any case, obviously. So they send up this clock into space in a microwave. I'm going to call it that, clock in a microwave.
Is it cooked yet?
And they took a while to cook.
Center's cold?
Yeah, the center's still cold. It's like a hot pocket where the inside is just, it's still frozen.
The pocket's cold. Cold pocket, hot
clock.
Sleeve?
And so they sent this clock up into space to see how accurate it would be. Why is this important? Now, when you are sending stuff into space, it's important to know where it is. So if you're sending something to Mars, you need to hit it pretty precisely because you're traveling millions of kilometers, and it has to land in the right spot. It has to hit the orbits, and you want it to be over the right spot so you can drop stuff on the planet. So you use this by checking the clock on the spaceships. You send a signal out to the clock, out to the whatever vehicle that you've sent out into space, and then you get a reception back of how long did this time take to go out there and back. When you receive it, you can tell how far away it is, right? And you kind of get a gist of we sent it this way and we got it this length of time, so that's how far away it is. Sort of like sonar? Yeah, kind of a little bit, except for it has the information in it. It's not just not just how, a little bit. You check the delay between the two. And that tells you how, and you get, the more accurate that clock is, the more accurate the position. Does that make sense? Sure. So if you know to a good degree of accuracy, you know to a good degree of accuracy. If you know to a good degree of accuracy when it is, you know to a good degree of accuracy where it is, which is why clocks are so important in space travel and information relay. But isn't time relative? It is, but you can correct for that. So if things don't really travel at relativistic speeds, even in space, it takes a lot of energy to start really dilating time. I think if you spend a year in space, you've only gained 6 microseconds on the rest of the people on the planet. So it's not a huge factor. But this clock that they sent up, they finally compiled the results and published how accurate it is. Okay, let's give us some deets. They were looking to make a box that would be within 300 microseconds. What's current? 300 0.3 microsecond. What's our current accuracy? So super accurate, picoseconds, trillionths of a second. We can keep things to. They sent this up to. They were hoping to get 0.3 nanoseconds. I want to get this right. 0.3 nanoseconds. So 300 picoseconds. And the clock that they made, the accuracy turns out to be 26 picoseconds. Whoa. It is an incredible, 26 picoseconds per day, it would drift off of what they had initially set it to. So with that kind of accuracy, you can get very accurate measurements. And with that kind of accuracy, you don't need to send a signal back to Earth. So if you send a signal from Earth to the ship and then get it back, that can take a long time to kind of figure out where it is. And the further out you go, it's hours if you're going into deep space. Days, Voyager, I think a round trip is 2 weeks at this stage. With a satellite that has its own clock, you don't need it. It can do its own accuracy. And so it can take pictures, it can keep its own timekeeping, and it can say, I know how far I've traveled over this much time. The more accurate those two measurements are, the more accurate your precision of location can be. So this new clock, it can really change the way we do deep space travel and the way we build our deep space satellites. Did they call it the super clock? They should have. Does it have a name? Yeah, it's the DSAC or some boring acronym. Yeah. DSAC? Yeah. Deep Space Astro Clock. That's my guess. That's as good as mine. It's really disappointing. The DSAC. I don't know if they meant. I probably got it wrong. It's probably not the DSAC. That's regrettable. It's unfortunate. All right. Well, that's my fact. A very accurate clock was sent into space. Congratulations, clock. Yeah. I can't wait for the new iPhone to feature the DSAC. Cool. Okay. Well, my fact, Vinny. You ready for this? I'm ready. I'm super ready. We're talking hybrids. The car? No. The organisms. What do you know about hybrids? What is a hybrid? Oh, like a horse and a donkey making a mule? Nice. So what is that? That's an example. It's a hybrid.
So hybrids are defined by two separate species
Right.
coming together and making an offspring.
Okay, like a liger.
Like a liger or a
Wolfen.
cayenne. Yes. And the thing with hybrids is they're really fascinating because sometimes they suck and sometimes they're awesome.
wait, why do they suck?
Well, sometimes they're much weaker
Okay.
than either species.
Well, maybe they're
and they're not
smarter.
able to live to adulthood, and they often are sterile, even if
Okay.
they do live to adulthood,
Right.
like a mule.
Like a mule.
And often those are the good ones, or the pretty good ones, because oftentimes they don't even survive.
Not viable.
Right? They're non-viable, exactly. But sometimes they're even better than the real thing. And we
Whoa.
call that phenomenon hybrid vigor.
Oh.
And what's interesting is we've known of hybrid vigor for a long time, hundreds of years.
We all have.
But no one really had good ideas as to why. Why can hybrids sometimes be better than the two separate species?
Okay.
So this fact comes from a very recent study looking at corn. So plants of all organisms, there's a lot of hybrids going
Sure.
on, and a lot of them are very successful compared to animals where they're less successful.
Right.
And so they wanted to look at why is hybrid corn, which I guess they've observed, better, stronger, bigger kernels, all these sort of signs of better growth
Yeah.
than non-hybrid corn. And so prior to their experiment, they suspected it might have something to do with their relationships between the plants and microbes. So
microbes.
lots of plants have symbiosis, that's sort of a mutualistic benefit relationship with microbes either on the plant, fighting like infection, or in the soil especially. So plants, like all things, have difficulty, for the most part, getting nitrogen. Nitrogen's a big challenge for a lot of living things to get at. It's hard to get nitrogen from the air because of the structure of the molecule, even though it's around us. So stable.
So stable.
I wish it was as stable as N2. Am I right? And so they get microbes, bacteria and fungus that will live in their roots and be able to change that N2 into more biologically absorbable nitrogen.
Mm-hmm.
And so maybe that has something to do with it. So they did this experiment where they took hybrid corn and not hybrid corn and put it in, sort of observed its growth in nature versus in soil that they stripped of all microbes.
Oh.
And they found that when they put it in that stripped soil, they did equally well. Normally, hybrid grows way bigger than the non-hybrid.
Oh.
But if you remove the soil microbes, it evens the playing field. So that's strong evidence that at least that the microbes have some effect, positive effect on hybrids potentially, and can be part of the reason at least why.
That's amazing.
Now, here's the cool thing. So
Whoa,
the
that wasn't
question
the cool thing?
Well, that's part of it. It's not the whole story yet. So it can be one of two things, right? Assuming that this is real evidence that microbes are affecting growth.
Right.
It could be that they are benefiting the hybrid,
Yeah.
or they're having a negative impact
right.
on non-hybrids.
Mm-hmm.
And it turns out it is the latter. They don't know why yet, but for their results suggest that the microbes that normally exist in the soil have a negative impact on non-hybrid growth. It's not that they're helping the hybrids grow.
Right.
It's just that they are not having a negative impact on the hybrids. Why? They don't know. They suspect it might have something to do with the immune system, and that for whatever reason, non-hybrids are their immune system fighting the microbes. They spend so much energy fighting the microbes that has a negative impact on their
Whoa.
growth. But that still remains to be seen.
I also didn't know that plants had immune systems.
Oh, yes. All living
Very
things,
cool.
all living things have some sense
Some
of
form,
fighting.
right? I guess you'd have to have it.
Even bacteria can fight viral infections.
Can an immune system have an immune system?
Maybe.
Meta-immune.
Meta-immune system. Well, maybe our guest today can shed a light on this. I mean, the fact that they're dealing with, I think, some biotechnology suggests they might have some editing knowledge. That was my rough segue at introducing our guest today, who is Lizzie Baker. She's soon beginning her first year in medical school at Dalhousie University and recently defended her master's thesis in biochemistry. Congrats on that, Lizzie. During her master's, she bioengineered a new type of spider silk designed to promote the growth of nerves with hopes of developing new treatments for spinal cord injuries. Outside of science, Lizzie loves music, comedy, sports, and her Chihuahua son, Chai. Please join me in welcoming Lizzie Baker. Come on down.
Hello.
Hi,
Thank
Lizzie.
you for the intro. It was so nice.
Thank you for writing it.
Of course.
Welcome.
Welcome. Yeah, so Lizzie, we are going to dive right in.
Mm-hmm.
Yeah.
Spider silk.
Yeah.
Maybe before we get to your particular study, do you want to talk a bit about the field of bioengineering a bit, and then we'll get to spider silk, and then we'll get to your specific project?
Oh, yeah. So bioengineering, or even mine was kind of along the lines of biomedical engineering, but I know you're kind of interested about like how you can kind of like trick organisms into making things that aren't natural, which I also think is really cool. And so, yeah, it's a huge field, and especially for making kind of new treatments, new therapeutics. You hear a lot about, I don't know, like different peptides sequences now that they're trying to use to treat different things, or coatings for medical devices. And we can make all of those by basically telling another organism what to produce. We can like alter, like E. coli, for example, which is probably the main one that people use for bioengineering. We can tell E. coli to produce a certain protein by editing its genome. And when I started my master's, I had never really done that before. So this was news to me. But it's actually like, you'd be surprised by how simple it is now, because like once you're trained in how to do this, like it is so prevalent. It's so prevalent. So yeah, there's a lot of different things that are being done with bioengineering, especially in E. coli, even things for like the food industry. Yeah, it's very cool.
Wow.
So how do you trick, can you explain in very simple terms for us?
Yes.
How do you trick an E. coli, for example, to make something it doesn't have a gene for naturally?
Is it like a just for laughs gags?
Like, so I think of like E. coli as like a little blob. Because I think of like, I know when we're making this in the lab, it's trillions of cells in a flask. But if you take it down to like a single cell, like a little blob, we basically make, by heating up the E. coli cells, it kind of creates pores in their membrane, or like their shell around the cell. And with these pores, you can stick stuff in there.
Wow.
And then close them up, and it'll take it in as its own, and go from there. So basically you need a synthetic gene or synthetic DNA, which we call a plasmid or a vector. And then basically like a company can synthesize that from anywhere. Like you can make any synthetic gene. It's very crazy. So you design that on your computer, they make it, they send it to you, put it in the E. coli, and it goes from
And
there.
you're good to go.
It's very straightforward.
It sounds like
it or not.
3D printing, but for proteins or whatever you want to make your E. coli's do.
Definitely, yeah.
Disturbingly simple.
Disturbingly simple.
And a lot of that has to do, I guess, with the fact that the alphabet, right? The words we use in DNA are the same for most organisms. So it makes it pretty easy. If we give them the word to make, they know how to make that word.
Yeah.
You know?
Exactly. Like for the most part, they're using their own cellular machinery to produce your synthetic or recombinant, which is what I call mine, protein.
So maybe there's like an E. coli movie coming, like the Matrix for E. coli. Because they're all plugged into the machine, and then someone like there's like a neo E. coli who's like
self-actualization of E. coli.
Yeah, it's like you're not. This is not what you're meant to be. You don't live in the real E. coli world.
It's really sad when you put it that way, I guess. What are we doing, Lizzie?
I don't know. I one time heard a peer of mine thank all of the E. coli that were used in the making
You
of her
should.
project, which was
That's a good.
very sweet, I thought.
Yeah, I think
But
that's.
it's a
Yeah.
big sacrifice. So.
Yeah.
In all my theses, I always gave a shout out to all the, the studied organisms that gave their life for this chapter.
Nice. Very good.
So you were particularly interested in spider silk, or utilizing the fact that spiders make this material and to make them make new stuff. So you want to talk maybe a bit about spider silk and what makes it cool before we move on to using it for other things?
Yes. So spiders are really just one of many different animals, insects primarily, that make silks. Even things like bees and wasps are able to produce silks. And of course, silkworm, which is what most people are familiar with when you think of like silk as a textile. Spiders are of like a lot of interest to researchers right now because they produce 7 different types of silk. It depends on the species of spider, but the most we know right now is there's 7 different kinds, and they all have kind of their own key features. So that makes it a lot more tunable. If you need kind of like a stronger material or a more flexible material or a sticky material, there's different silks that have all of those properties. And so that's why I think they're kind of at peak interest right now for material applications. But for kind of my work, I'm more in the biomaterials and biomedical engineering side of the field. We're interested in them primarily because of their mechanical properties. So if you think of something like a Band-Aid, it has to be, strong enough so it stays on your skin and it covers the wound, but it also has to like move around with your skin. It has to be a little bit flexible and stretchy and withstand all those forces of just moving as a human. So that's why we're interested in spider silk for biomaterials specifically.
Very cool.
Yeah.
Now I know that like my silk shirts are made from silkworm silk.
Yeah.
Why aren't we building shirts out of spider silk?
That's a really good question. I would assume it's because we can't farm spiders like we farm silkworms. Like it sounds absurd to think of a little farm of spiders. I mean, it sounds weird for me too to think about worms, but I mean, you have a big pot of dirt. I'm sure you can just stick a bunch of worms in there. I'm sure that's like how it works to some extent. I don't know the logistics.
Spider Farm sounds like a Halloween movie.
But it would not work whatsoever because spiders are extremely territorial. They're cannibalistic.
Wow.
So they would have to have their own little cage and separate from all the other spiders.
Oh, man.
And then there's, species to species variability with all these different proteins. And what's interesting is one study actually found that when spiders are held in captivity, they produce lower quality silks and more variable silks, which makes sense. They're probably stressed.
I mean, that's probably true of almost any animal, I would imagine. Captivity versus wild.
Oh, yeah. Animals act crazy. Animals gone wild when you put them, ironically, in a non-wild situation. Like,
they're stressed.
They don't know what to do.
Like I worked with salamanders during my master's, and as soon as you put them in an aquarium and there's 2 of them, one will totally change its shape and become a cannibal, whereas it's very rare in nature, right? But they just freak out when they're in close quarters with other organisms and just start eating each other.
I guess I'm against spider farms.
Now you know.
But I mean, like, if you're studying spiders and spider silk, you have to have them in captivity in the lab. Are there strategies to get away from those sort of trappings of captivity?
Are they free range spiders?
So I think like that's the main thing that's forcing us to produce them synthetically is because it's so hard. And so there are, of course, labs that do have spiders in captivity, and those, that's where all the literature comes from. That's how we know that spider silks, the different ones, all their mechanical properties, that's how we know it's so tough, and that's how we can compare our recombinant silks to the natural silks. So I know it has been done, but I think it's like the main driving force for kind of what I was doing.
And so how did you do it? How do you synthetically produce spider silk?
Yeah, so for me, my silk, the version of silk that I was producing, we call that a construct, like a protein construct. And so my construct was designed by a former postdoc in our lab. Her name is Dr. Lingling Zhu, and she designed this construct of silk to have two additional kind of like sequences on either end of the protein, which were there to actually bind a molecule that helps nerve cells grow and would support kind of like the growth and differentiation of nerves. So that's kind of how the whole nerve stuff came into play with this protein. But she designed it to be like that. And then that's when I came in, was to actually put it into the E. coli, tell them to synthesize it. So that's called protein expression, is when you get the E. coli to express your protein of interest. And then from there, you have to purify it, because your protein isn't the only protein within those trillions of E. coli cells that are in a flask. So you kind of have to go from there to isolate just your one. And once you have a pure sample of that, you can kind of go about the experiments.
We're gonna break this down.
Yeah, I know it's a lot.
So if I have my blob of E. coli, when it's made the silk, is it actually like, if I were to cut open the E. coli, I would find a giant thread? Is it like actually in the thread, or do you have to produce the thread from it?
So the thread, like what you think of when you see a spider web, like the lines, those, we call those fibers, silk fibers. And so inside the spider, it's kind of the same as when it's inside the spider before the spider makes the web, right? So in that form, it's just a bunch of molecules. And they are extremely insoluble in nature, but kind of before they get to that point, they're just like floating around. And what the spiders do in nature is actually, it's kind of weird. It's like, because I'm sure you've seen a spider like dangling from its butt, its little butt, I call it, with the silk. So it pushes, that's a gland, it's not its actual butt, but it pushes the silk molecules through that narrow structure. And so the narrowing, and then also the, ions in the solution and the pH, they cause all the molecules to come together. And that's how it comes out as a string of fiber. But before that, it's all in solution.
Okay, so it's basically, spiders are like 3D printers.
Yes, they are for sure.
More 3D printer.
There's a lot of 3D printer stuff going on. I mean, well, that's what a spider is. It's like it takes the raw material and then it assembles it into this like fiber, and then it puts it into a geometric shape that it uses.
Yeah, very fancy.
Vinny's selling 3D printers.
Yeah, spider shaped ones.
So what would the shape of these proteins be? Would they be like globular proteins that get lumped together to build fibers?
Yep, they are. Like, our lab has done kind of all that fundamental research on the type of silk that we study, which is a aciniform silk, or wrapping silk, they call it. It's the silk that spiders use in nature to wrap up their prey. And they also use it in their egg casing as well. So that's the type of silk that we study. And our lab has looked at kind of like a single protein molecule and done structural studies to see like, what does this look like? And it is kind of just a blob. There are some regions that are a bit more rigid and some that are a bit more flexible, and they've kind of determined that those specific regions are what come together to connect the multiple molecules that eventually will be able to form a fiber.
Amazing.
Yeah.
Unbelievable.
And so we have all these E. coli in a flask. You tell them like, hey, make this protein, and then stick on these two extra parts that we're going to use. And then the E. coli are like, sure, you're feeding us, I'll do this. You told me what to do. And then they just go, and then out come these proteins, or they make it in them. And then and then you have to go in and get them out. The gory extraction process. And then you have to distill it. So you have to like I don't know, what's the distillation process?
Yeah, how do you separate them?
There's so many steps. And I think, from what I know from our lab in the past, is every protein you make is going to be a little bit different. So you're going to that's kind of one of the main first steps, or was one of the main first steps of my project, was to determine how to actually get this and a good sample, because I want to grow cells on it. So it has to be pure. And so the first step basically includes crushing the E. coli cells under extreme pressure so that they burst and release their contents. And then once you have that, you can kind of separate the DNA and cell membrane bits that you don't want from the other protein. And then once you have just protein, you can kind of work between soluble proteins and insoluble. And I think, as I mentioned, spider silk is very, very insoluble. Like it's not meant to dissolve, right? You don't, your spider's web out there in the forest, it's gonna rain, there's gonna be wind, it has to be quite indestructible. So that makes it a little bit easier, I found anyway, because there's not many other proteins that are that insoluble as my spider silk protein. So once you kind of have that separation, I then actually used a column. I'm not sure if you're familiar with that part of biochemistry, but basically for other people who aren't, it's like a tube, a long tube. And on the inside of the tube can be many different things, but in mine there were beads with a specific ion in there. And so when I put my protein sample through, my protein was able to bind to it. So then all the waste would come through the end of the column. And then my protein, I know, was like isolated in there. And then I could just elute it off the column, and that's when I had my pure sample.
Amazing.
And so how much would you get out of like a single run?
So I would do the biggest cultures of E. coli I could possibly, because I needed this for a material. So I would make 3 liters of E. coli culture. And from 3 liters, I would get, the most I ever got was about 85 milligrams.
Wow.
Yeah.
And how long does that process take? From like starting your culture to like having it in your hand?
If I really pushed, I could do it in a week. Okay. But it is long days of work and getting splashed with E. coli. And I would typically do it over 2 weeks just to like stay a bit more sane. Yeah.
And so once you have all your globs of silk
the
purified,
technical term.
Yes.
Globular protein is a thing.
It's a thing.
It's a thing. See?
Okay,
professional.
Okay.
How do you force them to? You don't have a spider butt, so how do you force them together?
We were making a lot of assumptions here.
Yes, you may or may not have a spider gland, but how do you build the fibers now that you have the globs? Just poke them with the stick. Like, how do you fiberize?
That's your first instinct.
Yeah,
it's a really great question, and a lot of work had to be done to figure this out. So this is kind of one of the master students who came before me. This was his project, was how. Because they did find they could hand draw them. So like you have a little tube with your solution of silk in it. And if it was a high enough concentration, those molecules would interact on their own, like a self-assembly of sorts. And so you could stick a needle in and hook a silk fiber if you got lucky and pull it out. And that's what we call hand-drawn fibers. But of course, where there's not that pressure of the silk being extruded through a narrow opening, we found that, or they found, I should say, I did not help with this at all, but they found that kind of the mechanical properties of those fibers weren't as good as they could have been. And so that's kind of what pushed to kind of make a synthetic apparatus to form fibers. So then what he did during his master's was, it's basically like a syringe with a needle, and we put our solution in the syringe, very high concentration of the protein again. And then it's all, I'm not good with like machine stuff, but it's like mechanically operated so that it's pushing down at a constant force and a constant rate. And then it extrudes the silk through the narrow needle, which forms a fiber, and it goes into kind of a coagulation bath, which is something like ethanol, which further forces the protein together. And then you have to put your hand in the bath to pull out or hook it on something to pull the silk fiber out. And then we have collector tubes that are like wheels spinning around. So once we have the silk in our tweezers, we can wrap it around the wheel. And then hopefully, if you're lucky, it keeps spinning and collecting it. But often it might break, or it's not a flawless process.
Wow.
So the syringe is like a fake spider butt.
Yeah.
Wow.
Yeah, and we had to, of course, do a lot of research into kind of what solvents to put the protein in that would kind of mimic that environment to spider butt. Yeah.
How do we artificially recreate a spider butt? That's It's
I want to
important.
read that paper. This is so super cool.
Mhm.
I have a bit of a lateral question, and you might not have touched this at all, Lizzie, but I teach a couple biology courses. This is not my specialty, but I know one of the things we throw out at students when we talk about bioengineering is that it has occurred that people have used goats to make spider silk in their milk. And so that's like a way of making a bunch of spider silk the way that they make milk. They make spider silk instead, and then they extract it from the milk.
Spider
so when
silk
you take
milk.
the milk from the goats, you have a bunch of silk. Okay, that's not something you know anything. I was just gonna say, like, is that that seems like an easier way to get a bunch of silk, but I don't know anything about this.
I mean, if you think about it, it's kind of the same idea. It's like you're squirting it out of a small hole.
Yeah.
And there's, inside the goat is this solution of stuff that you're squirting out. But I did not come across that, I find that hilarious. That is wild sounding.
Maybe I had a fever dream. No, it is a thing. But maybe, I'm sure it's harder than it sounds.
You have to get like a spider and a goat to fall in love and go through a courtship process.
And that's hybrid vigor.
Oh, yeah, I get it now.
Yeah. Interesting. So now let's move to the other side.
Yes.
We'll talk a bit about nerves and the difficulty in having them grow normally.
Yeah, what's the big deal with nerves?
And then we'll connect it to spider silk. Now, what's what's up with nerves not growing themselves?
Nerves, they're just, they're not great.
Nerves are listening.
That's what I'll say.
Bottom 5 cells.
Yeah. And I mean, it's still kind of a mystery, really. Yeah. Because, and this is actually something I came across through my defense process with my thesis. I had a really interesting committee member who studies fish proteins. And so she brought up the fact, why can other animals do this? If, fish have some regenerative capacity. You think of things like salamanders or amphibians, they're able to regrow limbs that get cut off or something. So why can't mammals do it? And yeah, so the short answer is we don't exactly know, but there are a lot of hypotheses now that make sense, but there's never going to be one answer. It's kind of a combination of things. It's like the extent of the injury, what happened, your immune response that is mounted, kind of the poor environment to promote growth that is left following a nerve injury, the lack of kind of factors to stimulate growth in adults. So we have all these growth factors when we're developing, a little baby in the womb. We're growing our brains and things like that. So we have all those molecules to tell us to do that. But once we've grown and we've produced all these things, those kind of stimulatory effects, they go away because we're not supposed to need them as adults. And so without those, it is really hard to regenerate nerves following injury. Yeah. But it also has a lot to do with kind of the negative environment that's left in the spinal cord following injury.
What makes it a negative environment?
So a lot of people, I think, are familiar with like the blood-brain barrier. So it's a very separate, like the inside of your brain is very separate from the outside. So you have a spinal cord barrier as well. So when that's ruptured, it's kind of just this disruption of homeostasis. And so all these things are coming in that aren't supposed to be there. It's just not a great environment for growth, and all these inflammatory cells are coming in.
It's like blowing a hole in a clean room.
But so mammals as a whole cannot make new neurons. Is that true? No, like that's a whole mammal thing?
For the most part, from what I know, yes. We have our central nervous system, brain and spinal cord, and then we have peripheral nervous system, which is your limbs, the rest of your body. And that peripheral nervous system can, to some extent, depending on the length of injury, like the gap at that site and the type of injury. There's so many different types, and nerves are so complex, like anatomically. Like I think when I started this process, I thought of a nerve as just kind of one long tube of a structure, but there's so many layers within that tube and all these things, components that come together. And so it's like what parts are actually broken, um, and based on that, it determines like if it's actually able to regenerate.
And so, how does the spider silk come into this?
Yeah.
Hmm. So, as I mentioned, kind of like if you injure a nerve, there could be a gap between, you know, the end, one end and another. And so the difficulty is with the nerve cells or the axons, which are those long branch-like projections that come out of a nerve cell body. That's where the difficulty is, like the ability for those to extend over the gap to then reconnect at the other end. And so we're thinking if they had something to kind of help guide them there, like a material, a support system, a scaffold, if you will, to kind of facilitate the navigation through that poor environment, which I just described, to hopefully kind of reach the other end and bridge the gap. Yeah, so we're thinking spider silk could be a material suitable for that.
So the spider silk would be sort of between the neurons?
Mhm.
Interesting.
Yeah, or there's actually materials on the market for nerve injuries right now, which are called guidance conduits. And they look like a hollow tube, like a cylinder that's hollow inside. And largely they've been made out of polymers or collagen. A lot of research has been going into things that mimic the extracellular matrix, which is kind of the outside of the cell, so the outside environment which the axons would have to grow through. So they have tubes made out of that. And so something I kind of have thought of as well is if the inside of those tubes were kind of lined with spider silk for additional support to kind of make a sort of 3D matrix for cell growth. Yeah.
And these would be mostly used in any nerve cell, specifically for the spinal cord, specifically for the brain? Like, how interchangeable are these nerves?
So there's a lot of differences, of course, depending on where you are in the body. The guidance conduits that are kind of out there right now, they're largely used in the peripheral nervous system, so things like the sciatic nerve. And once again, it depends on how long the gap is at the site. But my project was kind of more specific for the spinal cord. So that's what I focused on, was mainly the spinal cord. Yeah.
So do you know at this point, I guess this is probably early on in this materials learning, but do you know how would you give this to someone? Would they take a pill? Would you squirt it? How would it be applied?
So it would be like a surgical intervention. It would kind of be like an implant is how I like to think of it. And if it were just the spider silk, like maybe like more of a bandage type thing, or just like silk fibers on their own that could be like implanted into this site somehow. The thought is that it would eventually degrade over time.
Right.
So it's like you put it in as an implant to help act as that scaffold for the axons trying to extend over the gap of injury, and then it would go away on its own, and the body would degrade it eventually. So that's kind of the overall idea.
Wow,
this is very cool.
It is very cool.
I think we could ask you questions all day, but we also want to talk about little Lizzie. Were you always interested in science and biology?
Were you bitten by a spider?
Yeah. Are you Spider Woman? How did you get interested in science in general?
I think it didn't stem from the spiders. I'm actually terrified of spiders.
Like, really?
I make other people like kill them if they're in the house somewhere. Like, I do not deal with that.
That's hilarious.
Yeah. But just like I was interested in science in general, and obviously I'm interested in medicine, and that's kind of what led me to this specific project. So it was very like biomedical related. But for science as a whole, I think I was always interested in just kind of like the outdoors and being very inspired by that. I grew up on the south shore of the province of Nova Scotia, which is like so many, beaches and lakes and lots of coastline to explore. So I was always very immersed in that, and really just piqued my interest for like natural things. And that's what led me to science. And then I kind of found what I was interested in, what I liked, which was more towards the medical field. And that's how I got here. Yeah.
That's very cool.
Yeah. So are you hoping that well, I mean, it's early, you haven't even started medical school, but are you hoping to sort of tie in your research, or is this sort of a whole new pursuit for you?
It was initially a whole new pursuit for me. But the more I got into it and the more I learned about it, I was very, very intrigued. And especially, going to things like conferences, I got to talk with neurosurgeons and neurologists and kind of learn about those fields. So I think it could tie in the end. And something I've always been interested in with respect to medicine is pathology, which is kind of the study and diagnosis of disease, and which is one of the reasons why I took this project for my master's, was because I knew I'd get to look under the microscope a lot. So that's something I really love, and something like I took with me from this project. And so just kind of that analyzing things under the microscope, I think could definitely be applied to my future. And even there's such niche subspecialties of medicine out there, like neuropathology. I had never thought of that before, but so many options.
And had you always been kind of like intrigued by the very small? Is this like or is this like recent?
No, I've always been intrigued by that, but like I need to still be able to see it. And so that's why I think like the microscope, I was so intrigued by that, is because you can see tiny things even bigger. And it's just like visual proof. So many people get like, I don't know, turned off by science because they're like, I can't see this. I don't understand it. Like DNA, I can't see DNA. What's it even doing? And so that's why like pathology and things looking under the microscope, because like it's very visual and qualitative, if you will. So yeah.
That's
physics
cool.
Though too small.
Too small. It's very small. I mean, we're talking about picoseconds. It's very tiny and precise.
Very tiny.
Yeah, you can keep that microscope or nothing.
Yeah.
And you want to tell us a bit before we let you go, because sadly we sucked up all our time asking about your research because it was just so cool. But do you want to tell us a bit about your Chihuahua? He's very cute in the picture you shared.
He's my child. I wear a necklace with his initial on it. His name is Chai, but with the T. I don't know if you're familiar with piano, but it's Tchaikovsky is a Russian pianist. So he's quite the little man. He's a long-haired Chihuahua. He's 7 pounds, 8 months old. And he gives the best kisses, but really always goes for like nostrils or eyeballs. And his aim is really good. Like he will get his tongue up your nostril.
Maybe he's just thirsty.
You should have named him Nettie Pot.
Nettie Pot.
Oh yeah. Yeah, but that's him. Yeah, he's a goof.
And so you mentioned you're also a music lover. Is Tchaikovsky a sign that you're a classical music lover?
You're smart. I grew up playing piano and taking piano lessons, and Tchaikovsky, I always picked his pieces if I got a choice because I just liked them the best. But these days I kind of still play piano just for fun. But I listen to a lot of other music, more mainstream, if you will. But I do love music. And in fact, Chai's middle name is Jovi Jagger for John Bon Jovi and Mick Jagger. So he's Chai Jovi Jagger. And
his last name is Imagine Dragons.
That would be wild. Yeah.
It's hyphenated for both his parents.
Yes.
Well, thank you, Lizzie Dragons.
Yes.
That's pretty cool.
Yeah, it's pretty badass. It's a very cute puppy.
Yes.
Thank you.
Well, thank you so much for joining us.
Yes,
Lizzie.
I wish we could chat longer. This is very, very cool research. I mean, anytime you're talking about making an organism make something from another organism, it is always so cool.
And then synthetically using it to fix people. That's incredible.
Hopefully one day.
Yeah. Baby steps, right?
Yeah.
And you put one of those baby steps on the map.
Thank you so much.
Well, thanks again to Lizzie Baker. Good luck with med school. You're going to nail it. Can't wait to read all about your spider silk treatments in the future.
That was incredible.
Thank you, guys.
Thank you. Oh my gosh.
Yeah. It's got it all. It's got spiders. It's got silk. It's got E. coli. It's got synthetic spider butts. It's got everything I love.
It's one of those like bioengineering, like like the subcategory of bioengineering of CRISPR. It's one of those things where it's like you hear it and you're like, that sounds easy. It can't be that easy. And of course there are tricky bits to the process, but it's like, wow, it's wild that we can do these.
And we're at the stage where we're just like, hey, E. coli, make this, and then they're like, all right.
You bring up interesting point though about the rights and autonomy of E. coli to follow their own destinies.
They're in the matrix. We plug them into something and tell them to make something that they're not necessarily supposed to make.
But because they're not purported to be sentient,
don't have to go
you don't
through
feel
an ethics
too bad
board,
about
right?
them. You don't. You know, it's interesting. Like even for for anyone who's not familiar, if ever you want to work on a live organism, depending on the type of organism, you have to go through a lot of rings to get permission. Mm-hmm. And I know that amphibians, there were perhaps a surprising number of rings to jump through to work on them. Mammals is a whole other ballgame of difficulty, even for something like a mouse, let alone humans. Mm-hmm. But as you get lower on the perceived hierarchy, you get to a point where no one cares. Like invertebrates too, although that's starting to change. If it's like an invertebrate that's listed, you have to get permission. Listed, like not necessarily endangered. Like there's a whole the celeb, there's like a hierarchy of protectedness.
Who's who?
It's a whole thing.
Apparently. Wow. Yeah. But E. coli.
No one cares about
we fight them. We can get an E. coli infection. There's so much of it around.
But they're not now.
They're not inherently bad. No.
Yeah, they get a bad rap.
Yeah.
And they certainly stink. That I know.
We didn't ask what 3 liters of E. coli might smell like.
Oh, I know what a tiny amount of it smells like, and I describe it as the worst halitosis you've ever smelled. Like terrible bad breath.
All right,
That's what.
Well, I'm glad we didn't ask now.
It's rough. I can't imagine. I didn't want to know.
No.
There's you'd see green stink lines coming out from those vats of E. coli.
Comic book lines are so bad. Yeah.
Where sound comes when you open it up.
Well, that was very cool.
Very cool.
All right, well, that's another episode.
We did it. Vinny.
Yes, Katie.
Do you want to send our listeners out on our socials?
So if you're interested in following us at Learn Real Good, you can check us out at LRG Pod on Facebook, Twitter, and Instagram. That's LRG Pod.
You should do radio sales stuff. You have such a great voice and you go into advertisement mode.
Thank you.
Okay, I don't know who this is. Also, if you are a grad student and you're like, hey, I'd like to talk to these goofs, shoot us an email at learnrealgoodpodcast, all one word, @gmail.com.
Perfect.
All right. Well, thanks, Vinny. Thanks, Lizzie.
Katie.
Thank you, listeners. See ya.
See ya.